Welcome, everybody. Thanks for joining me. Today is March 8th, 2023, and I'm going to do another Wednesday webinar. The first thing I wanted to say is I put out an Instagram post the other day about my water method, how I prepare the water, and we got a lot of questions from that. So some of you probably have seen that. The main question was, are you kidding me? Do I have to do all that? And the answer is clearly no. As I said in there, a lot of the reason I do this is just because I like fiddling around with water and seeing it go in vortexes and spinning it and all kinds of things. So for me, it's just sort of fun. But you don't have to do that. The best, easiest solutions are to filter the water somehow, a really good carbon filter or possibly reverse osmosis. Then put a teaspoon of the sea mineral, sea plasma in the water and stir it with an analemma wand. I think if you do that, that should be good enough for just about anybody. And I know people have a lot of questions about the analemma wand, so you can go and watch the podcast that I did with Dolph Santiga about 2 years ago, and we just booked him for another podcast, he and his research manager. So we will hear about all the goings on with the research they're doing into water and how to make it more coherent and the effect that coherent water has on plant, animal, and human health. And for those of you who have questions, it's good to write them to the customer service, and I will try to ask Dolph. We're happy to accept penetrating questions. That's what it's all about here, as everybody knows. The other two questions I got on that Instagram post about the water was, where do you get that funnel? So I showed you that I take the water from the Ophora water station, and it's in a big sort of, whatever pot that pours. I don't know what you call it. My mother used to call it like a big urinal. It's not a urinal, but you know what I mean. Anyways, I take that water and I pour it through this glass funnel, and it has very particular dimensions that were essentially given by Victor Schauberger, and then his grandson made it into a company that makes them. And they make acrylic ones, which are made, it's either Germany or Switzerland. And I don't even know the website because somebody got it for me. And the glass one was, I guess you could say, reverse engineered by a friend. And I just heard from him a while ago that he will consider, or at least price out, making some more of the glass ones, because I don't think it's easy to do. It's sort of like hand blown or something like that. It has to be the proportions, and the funnel part has to be just right. But we will look into making more of those, and then maybe we can carry them. But right now, otherwise, I don't really know where to get that. And the egg came from a dancingwithwater.com website, and they have these orgone plates that have copper rings around them, and then it's a ceramic egg, and you put the water with a little bit of the sea plasma in the egg, and you just let it circulate for a day, and that's where I got that from. Again, it's not like I've done serious scientific tests on all these things, so I can't go down that route with you. But certainly it makes sense to me that water needs to be moving somehow. It needs to be vortexing somehow. It needs to have some minerals in it, and it needs to be cleaned of fluoride and chloramines and birth control pills and whatever else may be in there. So that was the water thing. The next thing I wanted to do is I got an inordinate amount of people sending me a recent interview that was done with Derrick Broze and Peter McCullough. And it's really mostly going over the same thing, but there was something new in there which made me think that it's worthwhile actually going through this, because he brought up a new argument that I must say I hadn't heard before, that he claims proves the existence of the virus. And I thought it would be good to put that one to bed. By the way, just to be clear, after I saw this video, I wrote to Derrick and Peter McCullough and asked Derrick, as you'll see, he suggests we do more tests. And I asked him which test he thought we should do. And I haven't heard back from him. And I wrote to Peter saying it would be good to have a conversation about this, which he agreed, but said he doesn't really have time right now. So it's sort of on hold. So my thought was to facilitate that, I would essentially record, like I'm doing now, what I think about what he said, and then maybe that'll make it easier for him to either respond in writing or with a video, or it would help him prepare for our conversation if and when that actually happens. So that's a little bit of the background here. And with that, let me play the clip, and then I can tell you what I think about it.
Opinion on another topic that's related to COVID that has become the hot button issues in some corners. I'm sure you've come across it, but folks who believe that there are no viruses, or particularly that the COVID virus hasn't been isolated. I'm not sure if, how much time you put in or energy into that. I've interviewed Andrew Kaufman and some of the folks who are kind of promoting that idea. Personally, I'm not 100% sold on this idea. I think there's some research needs to be done. I do think there's some interesting data out there about FOIA requests that have been put out trying to get governments, can you provide me proof of isolation? But in general, what are your thoughts on this? Is this distraction, division? What do you think about that topic?
I think it's distraction, and it may even be intentional distraction. There are standard virology lab techniques that have been used for decades. Viruses are transferred into one cell culture versus another. They're isolated in order to be able to make vaccines. So of course they've been isolated. We can see them on electron microscopy. So we can actually physically see the viruses, and we can basically determine the entire genetic sequence of the virus. We can understand every single protein within the virus. So the viruses clearly exist. They've clearly been isolated because we make vaccines out of them. If they couldn't be isolated, we could actually never make a vaccine. The Chinese actually have the Sinovac coronavac vaccine is the isolated SARS-CoV-2 virus killed and given as a vaccine. So these claims are they're not useful claims. I don't think they're helping us get to any solution. And they're just, I think, distractions of people who just honestly don't understand standard virology and vaccine
when
techniques.
Someone says, this is one of the arguments I've heard, when their argument is when you look into the word isolation and the way virologists use it, they don't use it in the same sense that, so if I say I'm going to isolate the coins out of your pocket, all I have in my hand is coins, and they're saying that the process that's used to isolate viruses is not as clean cut as that, and that there's other material in there. And so this is their argument. Would you say that comes from a place of total lack of understanding?
Yeah, it's a lack of understanding. No, they're clearly isolated. I mean, the viruses are isolated, and it's actually purified in order to give us a vaccine.
Okay.
So they have to be isolated.
Okay, well, thank you. Thank you for addressing that. I just wanted to get
you. Okay. So I made a little bit of mistake here. Derrick Broze did not ask for more tests. He called for more research so that he could verify that the no virus so-called claim was accurate. And so again, I asked him what research or testing he would like to see, and I haven't heard back from him. So as you heard, Dr. McCullough made the claim that I hadn't heard before, which is that the Chinese are making vaccines. These, I'll tell you in a minute how they're making them, and that this proves that the viruses have been isolated and in fact purified. So even though in all our requests and all our looking at papers, we've not come across one example of a purified pathogenic virus, including SARS-CoV-2. So maybe Dr. McCullough can send us the reference of showing us a purified virus. But again, we've gone over the electron microscopy evidence for the virus. We've gone over the sequencing of the virus, and we haven't gone over this new claim that because the Chinese are making a vaccine of SARS-CoV-2, that must prove that the virus has been isolated and purified, or else how could they possibly have made the vaccine? So let's take a look at this claim. So I pulled this from somewhere, but I think it's sort of standard stuff, so I think we can basically rely on it because it's pretty much accurate for the standard response. So are all vaccines the same? So the answer is no. And then they go on to say the Chinese vaccines, which is the ones he's referring to, from Sinovac and Sinopharm, I'm not sure if it's Sino or Sino, are the main ones using this platform. This platform means they're using an inactivated vaccine because it, quote, contains a dead virus. The virus is still whole. It has all its parts in the correct shape. It can stimulate a response from the immune system, what we call antigens. The immune response can be against multiple antigens. And so that is the platform that he's referring to. It is an inactivated viral vaccine. They say it's a great technology. It works for human and veterinary vaccines used for the seasonal flu vaccine some years ago. And then they go on to talk about other types of vaccines, so we're not so interested. And then, of course, there's the obligatory computer pictures. So then we get down to the important point, which is how do you make these vaccines? And I'm going to read most of this. It depends on the platform. So we're not talking about the viral vectored vaccines. But let me just go over this because they say it's the same, for inactivated vaccines, the process is similar. So then you'll have this bulking up of the virus over the course of a few days, anywhere from 4 liters of cell culture to maybe 20 to 30 liters. Really high scale production can be carried out in steel tank. The manufacturing environment can look a bit similar to super clean, sterile brewery. You have to make sure that your cells are in the best environment possible for them to live and to allow the virus to grow. This requires monitoring many environmental factors in and around the cell culture: temperature, oxygen, CO2 levels, acidity, and so on. You end up with this liquid that is full of the virus you're interested in, but it's also full of materials you don't want. So then you have what we call downstream processing, where you're purifying the virus vaccine away from all the components that you're not interested in. This downstream process is very important and highly controlled and evaluated. It involves a lot of filtration and chromatography. At the end, you have a very safe and sterile product that contains only what you want. There are multiple steps, and at each step, you're taking samples and running experiments to show that you're purifying your product as you go along. Even though it can take a few days to grow a batch of virus, it can take a long time to purify it, and it's pure, sterile, and that's what you say it is. The vaccine will only be released when you can prove that it's the exact purity, sterility, and composition you're claiming. So here we get to the inactivated vaccines. The process is similar. you grow up liters of the virus itself, and then you kill it in a specific way so that you maintain the structure of that dead virus. And then you take that and you inject it into people. So again, you grow liters of the virus, then you kill it in a specific way. As far as I can tell, the two usual ways that the, quote, virus is killed is by heat inactivation. In other words, you heat it up, or they use a chemical called formaldehyde, which they say kills the virus, but it maintains the structure of the now dead virus. And then you take that brew, that culture material, and you inject that into people, sometimes with some amount of filtration or centrifugation or so-called purification. Now, let's go through these steps again. And the question that I want to ask is, at which step in this process did the people who are making the inactivated vaccine, A, prove there was a virus in this, and then prove that it was the virus that is growing in their cell culture? That is actually the only question that we're interested in right now. At which step, which part of this method was there the proof, or even I would say the possible proof, that you're dealing with an actual virus? So let's go through all the steps very clearly and methodically with those questions in mind. Which step is showing us the virus? So they take a person who is sick and they say, this looks like whatever illness they're talking about. In this case, we say that they have COVID. Now, you could say that the proof that they have COVID is because we all know at this point that COVID has no particular pathognomonic symptoms. Let me just show you that just to make sure everybody is on the same page. These are the symptoms of cold, flu, COVID, and RSV, and you can see they're basically identical. I won't spend a lot of time on this. Here's another one that says, from the CDC, no particular set of signs or symptoms can reliably discriminate COVID-19 from other respiratory viral illnesses such as the flu. So there is no possible way by looking at a person, examining the person, that you can say they have COVID. Even if you could do that, which you can't, that certainly doesn't demonstrate that the reason they're sick is because they have a virus. I certainly hope everybody would agree with that. All you know at this point is this person is sick with a non-specific respiratory illness. Okay, so then you take a sample of liquid or fluid from that patient, either a bronchial sample or mucus from their nose or maybe something else, but those are the usual ones. And let's look at that. So there's no examination done on that specimen. So there's no possible way that could show you that there's a virus there, because actually nothing is investigated. So then they put it through some, I would say, not purification steps, but they clarify it by putting it either through a filter that filters out the dead cells and the bacteria. And so all you have then is whatever is liquid from the person's mucus or lungs. And I would think that there's nobody who knows anything about this who would say that is a purified virus, or it even shows you the existence of a virus. Sometimes they do a different clarification process, which is called centrifuging it. Again, not looking for a virus, but just to get rid of the big cell, the cells and the bacteria. And then they have the supernatant, the liquid part. And importantly, and this is a crucial part of this analysis, there is no test done on this that could demonstrate the existence of a virus. They might do a PCR test, which is not a test. But we have to remember that these are PCR processes that can never show the existence of a virus. And the PCR process that is being used for SARS-CoV-2, we all remember, was made by Christian Drosten, who said, quote, we made this PCR without having access to any viral material. So nobody could possibly claim that the PCR examination of this centrifuged or filtered fluid could possibly prove the existence of a virus. There is no ultracentrifugation done at this step. There's no electron microscopy analysis of the fluid. So we have no idea that whether or not there's a virus, a particle that you could call a virus, in this supernatant or filtered fluid. And importantly, nobody at this point is looking for a virus or claiming that somehow these steps have found or demonstrated the existence of a virus. So that should be clear. So now let's say they filtered it. So we have all the liquid parts that come from the mucus or lung fluid of a sick person. We don't know why they're sick. We haven't seen any virus. We have the liquid, which contains probably hundreds, maybe more types of things. It has proteins, nucleic acids, minerals, lots of maybe poisons, toxins if they're in there. Lots of things are in there. I dare say nobody would claim that is a pure virus. So they take this fluid and they mix that into these big vats that contain cell cultures, mostly some type of vero cells. Then they add antibiotics like usually gentamicin, antifungals like amphotericin, both of which we have presented papers showing both of these are toxic to kidney cells and other types of cells, therefore could be the reason for the breakdown of these cells. They change the nutrient blend, and they also add fetal calf serum to this. They change the temperature a little bit and maybe the pH, so they add some other chemicals. And then to this, they add this mixture of many different many different substances, which may or may not include a virus, but the virus has never been seen. Now, if you're doing a scientific experiment, you, as we all again know by now, you have a dependent variable, which is the effect you're looking for, which in this case, then you're looking for, do these cells die? That's called the cytopathic effect. And then you're testing an independent variable, which is meant to be one thing that you're trying to investigate whether it caused this effect that you're looking for. So if we're trying to prove that only a virus caused the death of these cells, only the virus grew in this culture and caused the death of these cells, then by definition, the virus would have to be the independent variable. But in fact, what is the independent variable here? So the independent variable is a combination of antibiotics, change in nutrients, and all the things that are soluble from the bronchial fluid of a sick person. There is at no point up till now any even attempt to establish that there's a virus. All we can say is that some component of that mix, the soluble part of what's in somebody who's sick, the antibiotics, antifungals, change in nutrients, fetal bovine serum, some part of that broke down the cells, made it so that these broken down cells created essentially cellular debris, which, as we've said over and over again, are then misinterpreted as viruses. So the cells break down into all this debris. No attempt is made by these Chinese manufacturers then to identify any virus or prove that any virus is in that vat of broken down now cells, antibiotics, the filtrate from the person who is sick, etc. No attempt. They put that into vials, and that's the vaccine. So the question for Dr. McCullough is, which step in there proved the existence of a virus? Which step in there was the so-called isolation of the virus? Now, let's define isolation. As Derrick Broze said, isolation means to take something out of its environment so that you only have that single thing. If I have a bunch of things on my desk and I take the pencil, I have now isolated the pencil and only the pencil from my desk. In which step up till now did they, quote, isolate the virus? Because as far as I can see, there was, not only did they fail to isolate the virus, at this point, nobody has even attempted to demonstrate there's even a virus in this process at any point in the process. The importance of this is, if you haven't isolated and therefore seen and proven the virus to exist, any further evaluation, such as pictures with an electron microscopy, or evaluating parts of it like proteins or nucleic acids, you have no idea the origin of those nucleic acids, proteins, antibodies, or anything else in there, because at no point in this process did you obtain a pure sample. So let's be very clear what we're asking you. We're asking you to present proof, evidence, that at some point in this process, you have obtained a pure virus. You've seen it on an electron microscopy. There's nothing else in there but the virus. You've proven that that virus came from the original person. You've then proved that all of the nucleic acids come from that particle which you have purified, that there's no chance those particles came from the cells or the fetal bovine calf serum or anything else part of that mix. That's what we're asking you, not whether they say they isolated it, not whether they say there's electron microscopy pictures, not whether they say that the PCR proves that there's a virus, even though they got the PCR test essentially without even having an isolated or purified virus, which is their own words. We are asking for validating the methodology of that vaccine production process, which you stated should be considered proof that they isolated the virus. I'm hoping that this is very clear. And in any future discussions we have about the existence of the virus, it has to start with A, did you find the virus in its natural ecosystem? The answer, of course, is no. And then if you isolate the virus, as you say, through the cell culture process, how did you prove that the virus existed in the first place in order to do an experiment with it? And how have you proven that the cytopathic effect could have only come from the virus? Because every experiment that we've looked at has shown just the opposite. So hopefully that is very clear, and I can either wait for your answer either from a video or written, or hopefully you can look into this and then we can have a discussion as I proposed in my email. Again, I hope this is very clear, and I hope it's very clear that just saying an inactivated vaccine proves the existence of a virus is a scientifically unfeasible position. Okay, so I think we got through that. Now, some people say to me, which is, I must admit, partly true, maybe even largely true, that Tom, you never say a good word about what anybody does. You know, you're always criticizing people and this and that. So I was sent an analysis of what happened in northern Italy that was so-called COVID. And I want to go through that because in my opinion, even though, as I've said over and over again, to show that there is no virus, it's not our responsibility necessarily to say what caused certain people to get sick or not. The issue for us is that if there is no virus, then all the things, COVID obviously can't exist, and treating people based on their first infection or their second infection, or treating them with antivirals, or any strategy that looks at vaccine immunity or whatever, that's all out the window if there's no virus. So we're looking at the beginning of this. But this guy named, I think his name is Michael Bryant, wrote a very good paper that I thought I would share with you, looking in, which is really how a investigative reporter, I'm not sure that's what he is, should actually go about trying to figure out, so what actually happened to those people in Italy? So let's go through that. So I don't know where the link for this is, but we'll try to get it to you. If not today, then a certain point. And I got permission to share this, so I think we're good at that. We're good. So what happened in Italy 2020? And I'm just going to go through this because it really stands for itself. You know, the Western world came to a standstill. The narrative depicted a strange, suddenly super spreading, deadlier than flu virus hailing from China that landed in northern Italy, discovered in the West in the Lombardy town of whatever, Codogna. Later that day, the Italian government reported their first COVID-19 death. That's interesting. And then dramatic media reports emerging from northern Italy were hammered into and onto the Western psyche, giving the impression there was a mysterious, super spreading and super lethal novel virus galloping across the region, infecting and killing scores of people. Harrowing reports out of Bergamo, a city in the Alpine Lombardy region of northern Italy, spoke of coffins stacked high, COVID-related deaths growing relentlessly, and the alarming need for military assistance to remove the grim volume of dead bodies piling up. In early March 2020, hospitals in northern Italy were reporting a tsunami of deaths due to the COVID crisis and overcrowded conditions due to fighting the coronavirus outbreak, which were pushing hospitals and staff to the breaking point as doctors were taking the dead from morning till night. Using the entire machinery of the state, the prime minister began issuing a rolling set of government decrees, culminating in Italy becoming the first country in the world to implement a national lockdown. These mandates would set the stage for the lockdowns throughout the Western world. 3 years later, a comprehensive evaluation of the story about the alleged Italian medical emergency in spring 2020 reveals a tale of disturbing epidemiological history of northern Italy, mass media manipulation, and deceptive reporting utilized to create the illusion of a new epidemic. So the importance of that is this was no insignificant event, right? This was a huge and important event for the world as far as the whole COVID scam goes. So here are a number of questions surrounding this story. Were the overcrowded conditions in the Italian hospital generally the result of a unique viral pathogen, or were there other causal factors? Were these anomalous spikes in excessive death in northern Italy verifiably caused by the arrival and spread of a novel deadly virus? How is it that this virus spread across thousands of kilometers within days and peaked synchronously in selected locations? How was it that this virus was able to spread so fast across thousands of kilometers, peaking at the same time in these selected locations, yet wasn't contagious enough to spread to nearby locations? How was it that this virus waited for a government decree and only then began to create excess deaths? How was it possible that all countries in the West and beyond adopted similar health measures as carried out in Italy virtually overnight, measures that resembled a de facto police state rather than medical initiatives? Those are damn good questions. And so here's a timeline, and I won't go over that, but you can look at that as soon as we can find you the link. So what actually happened there? And I'll try to skim my way through this. First of all, he was able to document, and here's all the references here, that this area, I think they call it the Po River Valley, has the worst air quality in all of Europe, cited as having the highest mortality burden associated with air pollution in all of Europe, is known for its unique characteristics of low wind and prolonged episodes of climatic inversions, turning it into a holding tank for pollution. And then you go on to the planetary health report, talking about particulate matter and nitrogen dioxide pollution. And these cities had the two highest death rates, I think, of any places in Europe, especially the highest incidence of upper respiratory infections. They were also having ongoing and severe epidemics of all kinds of lung disease, idiopathic pulmonary fibrosis, interstitial lung disease, bronchial and lung cancer. They also had a history of years back of having exposure in the region in the 60s and 70s to occupational asbestos, and they were seeing this exposure as coming home to roost, so to speak, by a high level of mesothelioma, an aggressive and deadly form of cancer of the chest and abdomen, and it was peaking around this side. And they also had excessive flu deaths in basically all the years leading up to this, compared to other regions in Europe and around Italy. So they had also a lot of animal waste and milk production. They have poison soil. They had severe history of industrial accidents, probably something like the dioxin fiasco that we just had in Ohio. And so there was a lot of reasons why these people would suffer from increased respiratory diseases. Then he talks about the austerity and health infrastructure problems that particularly this area of Italy was having for years before this, maybe in some ways setting the stage for this. They say that more than 9 million people in this region declared that they could not access health services for some economic reason. That's not always a bad thing, but in some cases it can cause trouble. Further cuts in their healthcare budget happened in the years leading up to that. And the important thing is this resulted in a severe effects on the healthcare workforce and available hospital beds and equipment, essentially hampering the ability of care facilities to effectively treat people. And here you have some documentation of the decrease in acute medical units. And this becomes important when the claim is, so we don't have enough medical beds to treat all these sick people from COVID, but this turns out was happening before. So they talk about the number of ICU beds and the regular operational levels. 90% of them were occupied every winter during their so-called flu season. And they say that the flu outbreaks caused the units to fill up all over the world, but particularly here. And so all of this stuff was happening way before the onset of COVID. And then, of course, we have the new treatment protocols, which particularly are midazolam, morphine, and other drugs, which we now know were used during intubation. And this mechanical ventilation was being misused and overused, and we now know was being used to paralyze people and cause them to have heart trouble and even go into full-blown respiratory failure. Some of the doctors complained and tried to move away from using them. Here's a quote from an emergency room doctor who said that in the beginning we intubated everybody. Now we try to do this as little as possible. And they were noticing unusually high death rates for the patients they put on the ventilators. And so one has to ask, as he did, was this partially an epidemic of iatrogenesis, meaning a combination of iatrogenesis means doctor caused, but that was the final mechanism of essentially killing off all these sick people who were getting sick for all the usual reasons that people were getting sick. Then they talked about the conflicts of interest, which were of little interest to reporters. They talked about residents describing the abandoned streets, that food was not available, and all kinds of panic was happening in the region, supermarkets getting stormed. And it was, the mayor said it was difficult to contain the panic at first, especially as a lot of false news was circulating on social media that people believed to be true. There is still panic regarding food provisions, and people were trying to stock up on pasta and bread. And then there were all kinds of things like that happening. This is all, as he said, possibly manufactured hype from vested interests. And then you have the deceptive reporting. The media kept quiet on the fact that as recently as 2018, hospitals in Milan were overrun with viral lung infection, they say. And due to the aforementioned pollution problems, decimated health infrastructure, aging population, overrun hospitals have become a seasonal feature of the national profile of Italy over the last few years. And then, interestingly, there was a problem with the nurse workforce, who were apparently largely from Eastern European countries. And so they kept them from coming in, which exacerbated the healthcare crisis. Then, of course, they bring in the PCR, which is all false positive, but here they say even as high as 97%, but we all know they're all false positives. And then here's a quote from Stefano Scoglio, our friend. He discovered a new element of the real fraud, the choice to reduce the positivity to the swab by detecting only one of the three genes that would define SARS-CoV-2. If the virus were present, all three would have to be found, because if the virus is intact, in the only case in which we can have a pathogenic role and infect, the test must find all three genes. So apparently in that situation, they even changed the usual bogus test to say now you only need one of the genes, not three. And then they started manufacturing COVID deaths, and you see 99.2% of COVID-related deaths were people who had pre-existing chronic conditions. In other words, may have died anyways from the care that they received, so-called care or iatrogenesis. And there's a quote, "The way in which we code deaths in our country," this is a scientific advisor to Italy's health ministry, is very generous in the sense that all people who die in the hospitals with the coronavirus are deemed to be dying of the coronavirus. Only 12% of death certificates has shown a direct causality with the virus. I would say that's 0, but even with their own bogus way they report this, that's what they found. So even then, the mainstream press, they were reporting that all these deaths were people who were basically sick otherwise. So you can go on. Then he talks about the photographs that of the stacked coffins, which turn out to be one of them at least taken from a hangar in an airport back in October 5th, 2013. These were corpses of African migrants who perished in a shipwreck. And you can go into all the other fear-mongering and funerals and cremation anomalies that you can see. Then you get into the epidemiology, which is why it happened only in this one location, but in the neighboring regions of Italy, there was basically nothing, even though they had a larger concentration of Chinese people in other areas. Somehow, these areas weren't hit by the virus as much as the poor and impoverished and toxic and polluted and iatrogenic and crumbling infrastructure areas in this previous region. Okay, then let's finish with why did they choose Italy, besides all the reasons why you could manufacture a epidemic in that area with so many people getting sick. But they also talk about the economic catastrophe that was happening. This was a financially bankrupt country with a financial sector desperate for bailouts and a command structure run by central bankers made for a willing and compliant government. The citizens of Italy had been dubbed the sick man in Europe for the past decade by the EU financial sector. The Italian government was facing extreme economic pressures in 2019. They had officially slipped into recession in 2019, and they were worried that this was going to trigger a financial collapse all over Europe. By May 2019 was said to be posing major threats to the monetary targets of the European central banks. And then the tsunami of financial collapse staring East European central banks came to a head in 2019. And then they came in that the tried and true bailout scheme was proposed under the auspices or the excuse of dealing with the COVID crisis to the tune of 1.3 or 1.46 trillion euro bailout and a 750 billion euro recovery funds to deal with the COVID so-called crisis. So you can see a lot of reasons why this may have happened in Italy, and then in a later follow-up article or extended, he talks about even the role of electromagnetic fields and possibly 5G being introduced into the region and a bunch of other things. So let's just read the conclusion here. 3 years later, the indispensable truth of the Italian story is once you scratch beneath the surface of the official narrative of the COVID pandemic, it turns out to be a bottomless snake pit of distortions, manipulations, and outright lies. Any excess deaths in spring 2020 in northern Italy were an artifact of an already existing health conditions and an aging population, the obliteration of the existing healthcare infrastructure, massive industrial pollution creating chronic conditions, media-generated hysteria, savage government lockdowns, and administrative murder of the already fragile. These iatrogenic medicine-caused deaths of fragile people were the result of the social order and public health despotism and then used to give the impression that there was a, quote, deadly virus circulating. The only pandemic was one of violent government and biomedical assault against people. The evidence from Italy in 2020 exposed the official COVID narrative for what it is, a cold-blooded, organized deception. There was no pandemic. And all I can say is this is the reason why the virus story is so crucial here. As long as there's this bogus virus story out there and all we're going to do is look into the virus, and then just to give a shout out to the political circus now going on in Congress called the congressional hearings to investigate the origin of the unicorn. I mean, the SARS-CoV-2 virus, not the unicorn, not the invisible exploding unicorn. And like all political controversies, this one will never end, because how do you disprove or prove where the origin, whether it was a lab-created invisible unicorn or whether it naturally evolved from a bulldozer or a monkey or a horse? You never will get to the bottom of it, and that's exactly why they're doing this. And this is exactly why the health freedom community, which is essentially aligned to this narrative, they want to see this circus go on, people taking sides, slinging mud at each other, never getting to the bottom of it, saying useless, irrelevant, anti-scientific stuff, never answering the question that I asked in the first part of this. If this is what you call isolation, and everybody knows that in order to analyze something, you have to isolate it, have it in pure form first. Which step in this process isolated and has in pure form this particle you're calling SARS-CoV-2? Because if you never did that, there is no sense looking into the lab origin, natural origin, extraterrestrial origin, or any other kind of origin, because I will go on record as predicting this will never be settled. More and more people will write books, never asking the question, how do we know this thing exists in the first place? And it's just the usual useless, time-wasting, money-wasting political circus. And I hope everybody can see through that and let these people know that you're not interested in this circus. You're interested in addressing the concerns that Michael wrote about in this paper. Unless we get to the point where we demand or create our own world that's not polluted and not corrupt, and has a healthcare system that actually works for people, all the rest of it is just wasting our time. Okay, thanks everybody for listening, and we will send this to Peter and Derrick and hopefully get a response. And if we do, I will let you know. So take care, everybody, and I will see you next week.